As I said, it's the fifth one in our series. It's been an incredibly engaging and fruitful engagement with stakeholders from across the world. And I want to thank upfront our sponsors, Amgen, for creating this platform as part of the Bio-Africa dialogue. As you all know, Bio-Africa is a platform to promote dialogue in the African continent, in essence, creating an opportunity for Africans and for the global community, particularly the solidarity of the international partners, to structure dialogues that are intended to bring our views as well as our insights and wishes or points into the agenda for development of products. We started this conversation last year in the 2021 Bio-Africa Convention, again with the support of Amgen, and it was at the time quite a political conversation. And one of the fundamental questions that we were asking was, why do needs of the continent lag behind the rest of the world, particularly in biotechnology? We've always argued that biotechnology, unlike most of the other technologies or innovations, do not owe its existence to sunk investments, decades or centuries of previous investments, and that there's a real opportunity to use biotechnology in this continent to catch up with the rest of the world. Our conversations or dialogues for the past year have been really around how we mobilize the intellectual capital that resides in the continent and to use the international solidarity movement. And we've seen how COVID has really put that into practice, where there's been so much collaboration, but now we are seeing that, and particularly in today's conversations, we are really looking at the more advanced therapies like gene therapies, cell therapies, and other ways of doing clinical trials that could, in essence, ensure that the African continent and the participants in this continent are participating on an equal footing with the rest of the world, because the science is accessible to all. It's not an issue of infrastructure or resources. The science is accessible to us. And how do we use partnerships with the rest of the world to create a model or models that would move us forward? And I'm hoping and I'm looking forward to the 4 presentations from our panelists. And I'm not going to introduce the panelists at length. I'm just going to be requesting them to come through and they will make their presentation, and request them that they first introduce themselves, their institutions, and what they work on, and then they can take us through their presentation. And by way of call it house rules, we are not going to ask questions after each presentation. We are going to assume that the presentations are logically sequenced. And at the end of the presentations, I will then ask questions to each one of the panelists for about 5 minutes or so. And I would like to also invite the audience to type in their questions in the chat box, and those will then be asked directly to the panelists. Just by way of guidance, please indicate the panelists that you are asking the question to, and do not leave it to me to try and guess that, because I will mess it up. I can promise you that. It will be a catastrophe. So when you ask the question, please indicate Dr. Jennifer Ada, Dr. Janine Schofield, Professor Mushabela, or Dr. Ndembi, and then we'll take it from there. I think with that, I would like to invite our first panelist, Dr. Adair, who is going to speak to us about gene therapy, particularly focusing on an outsider's perspective. She's based in the US, and so she has a helicopter view, or rather a window view into what we are trying to achieve in this continent. Over to you, Jen. Thank you so much.
Thank you, Dr. Masumi. Just double checking that everyone can hear me okay. Great. So thanks to Amgen for sponsoring this lecture. And as Dr. Masumi mentioned, I'm located in Seattle, Washington. I'm an associate professor in the Cell and Gene Therapy program at the Fred Hutchinson Cancer Center here. I'm also a divisional member of the Translational Sciences and Therapeutics Division. And I've spent the last 15 years in the gene therapy space, bringing gene therapies from the bench to the bedside, so translating these into clinical trials, and that is the perspective that I'll be speaking from today. So genetic therapies are the use of genetic material to treat or prevent disease. And what makes gene therapy so attractive is that these types of approaches actually get at the underlying biology of disease rather than just management of symptoms. And that means that in some cases, a genetic therapy could be a one-time treatment with curative potential for otherwise diseases that may have required supportive care for the rest of the patient's life. So this slide is actually even out of date. I just sent it to the panel earlier this week. And the first bullet point, 22 approved gene therapies, is now no longer accurate. It's 23 as of this morning. As of 2021, there are 536 gene therapy developers worldwide. And as of 2020, there were 373 registered gene therapy trials. And the vast majority of this work has been developed in the USA, as shown in the pie chart on the right. Nearly 70% of the gene therapies that are currently in development or that have been approved were developed and researched and brought through the development pipeline in the United States. What's missing from that pie chart are countries that are defined as low or middle income by the World Bank, which are shown here in light and dark purple on this map of the world. And what I've overlaid on this map are these red crosses, and those represent countries that have homegrown gene therapy trials in progress. And India just became the first low and middle income country to actually have the development pipeline for gene therapy. And they have had the formation of the first gene therapy companies in India, including companies like Immunio. Why is this important? And what do we need to do to encourage the development pipeline in low and middle income countries? Well, one need look no further than case examples. And in particular, I'm going to be focusing on diseases that are prevalent in low and middle income countries. And so it seems it would make sense to allow gene therapy development for these diseases in the spaces where the majority of patients live. And the two case examples I will give are sickle cell disease and human immunodeficiency virus, or HIV infection. And I'll just abbreviate these SCV and HIV. These are two diseases that impact tens of millions of people globally, and 70% of the patient populations reside primarily in sub-Saharan Africa. Now, what makes these two case examples important is that there are already gene therapies in development in other parts of the world, again, primarily in the US and some in Europe, to treat these diseases. In fact, one of the first 6 gene therapies that was approved in the United States was a gene therapy to treat a red blood cell disorder called thalassemia, which could also be used to treat sickle cell disease. Between 2014 and 2021, there were 7 different clinical trials of gene therapy for sickle cell disease. And since the mid-90s, there have been nearly 40 clinical trials using some kind of gene therapy to treat HIV infection, which have registered, completed, or are currently enrolling. But the number of these trials which have taken place in Africa, where the majority of patients live, is exactly 0. So clearly we have a huge distribution gap, and importantly, we need to really think about, from the perspective of genetic therapies, which start with the patient's DNA and or genetic material, does it make sense for us to be developing gene therapies for these diseases which are prevalent in sub-Saharan Africa in places like Seattle, Washington, where I live? And in the last 10 years, the work of groups like H3 Africa and others that have spent time sequencing African genomes have indicated that that's probably not the best approach. And I'll highlight this particular paper that was released in 2020, where there was whole genome sequencing of 426 individuals across 50 ethno-linguistic groups among the African continent, covering a wide array of geographic regions. In those sequences, they found more than 3 million previously undescribed genetic variants across all other genomes in the world that had been sequenced. This unequivocally demonstrates the African continent's human population to be among the most diverse on a single continent on the globe. They also identified 62 new chromosomal locations that they found to be under what they called high selection. And those were involved in several pathways that are important for how we do gene therapy in other parts of the world. Viral immunity was one of them, and you could see where that has direct applications in the treatment of HIV. But the common method for delivering genetic therapies currently is actually using engineered viral particles. And so that viral immunity could play a big role in whether or not those therapies are readily taken up by patient cells. DNA repair was another important category. Again, this is important because DNA repair mechanisms often are the modality by which we introduce genetic changes or genetic material into the patient's DNA. And then finally, cellular metabolism, how DNA is converted to RNA is converted to protein and how quickly those proteins turn over. And if there are massive differences in those metabolic pathways, there may be very different outcomes in the gene therapy development process for different patient populations. So this is why in 2020, in collaboration with Dr. Sisikityo Mutuluza, the executive director of the Joint Clinical Research Center in Kampala, Uganda, we started what is called the Global Gene Therapy Initiative, or GGTI. The goal here is to enable the development of gene therapy for locally prevalent diseases, no matter where they exist, and primarily focusing on diseases in low and middle income countries where the gap is much larger. We published our first working group report in 2021. And in that working group report, we highlighted that there's so much more than just the technology to deliver a gene therapy and the genetics to understand how that gene therapy might play out. There are 4 pillars of activity that need to happen simultaneously in order to allow for clinical trials to have a chance of success in these spaces. The first is community outreach and education. This can't be stressed enough. If people don't know what gene therapy is or how it works or why it could be beneficial to them or what the potential risks associated with it might be, they're not likely to participate in a clinical trial. Clinical implementation and readiness, obviously, we have to have the capacity to do gene therapy locally. That means you need to build a workforce, you need educational programs and training programs, and you need to have the clinical infrastructure to be able to conduct the gene therapy clinical trial. Now, the way we do this in high-income countries is not the way that it's going to be sustainable, even in our own countries, let alone the rest of the world. And so there has to be a pipeline for new technology development that can encourage sustainability and affordability of these approaches. And you need a pathway for regulatory approval of these new therapies. And so you need infrastructure for commercialization. Therapies all around the world struggle to make it to patients if they can't have a commercialization phase. And you need public trust that this development process is going to make sure that benefits outweigh risks. And so you need policies in place at a government level in order to support that. Our group meets once a week and it's open to any membership. We meet virtually early in the morning for people like me on the West Coast, in the evening for people in Africa and Southeast Asia. And most of it is conversation to connect networks of people with expertise to help build each of these 4 pillars. And with that, I will stop and give it back to you, Dr. Masumi.
Thank you, Jen. I hope everybody enjoyed that presentation. And as I said, we will give the audience a chance to ask questions. And I'd like to welcome everyone again. I know we started a bit late, and I apologize again for the initial hiccups and the 5 minutes late start. Hopefully from here on, we are going to charge forward without any hiccups. And again, I'd like to thank the panel and thank you, Jennifer, for such an insightful presentation in terms of gene therapy. Gene therapy to me sounds like Star Wars or Star Trek. I hope what you've just explained, at least where I sit, it basically validates the reason why we have a Bi-Africa platform, among others, where we try to promote a global collaboration and an alliance of progressives, what we call an alliance of progressives, to work together as a single humanity, as opposed to different continents or different geo-regions and stuff like that. So thank you for that. And I'm looking forward to the questions and to the engagement between the other panel members. And I think without wasting much time, I want to move on to our next panelist. And this one is Dr. Janine Schofield. And I'm quite proud to announce that she is of the soil. So she's going to be presenting from the African soil. Thank you, Janine, over to you.
Dr. Masumi, and thank you so much for giving me the opportunity to speak with you today. I'm just going to make sure that my slides are up and running. I hope that they are. I'm sure someone will let me know. Naturally, first, just the disclosure from Amgen, but thank you very much to them for inviting me to join in on this conversation. So I'm a research group leader at the Council for Scientific and Industrial Research in Pretoria, South Africa, the CSIR. My work is predominantly surrounded on creating novel disease in a dish models to study how to improve drug pipeline development. But I also wear a second hat, and that is as the editor-in-chief of the Springer Nature journal Gene Therapy. So in tackling this problem, and I think Dr. Adair has put it together so beautifully. She's actually done half of the work that I need to do for me. But I guess when I think about this problem, I think about why we need the increased clinical diversity in clinical trials, and then start thinking about some of the ways where I think we can, how we can actually execute that in a resource limited situation. So I'm going to focus on those 3 ideas, why it's such a big problem. And again, Dr. Adair has touched on that, and what the consequences of that are, and then also how I think we can pull that back together. So I'm cognizant that with an audience like this, there are probably some pretty amazing geneticists here. And as a full disclosure, I want to be absolutely clear. This is a slide that I use for my students. But for those of you who are not familiar with the why we have such severe adverse drug reactions and how that's linked to a lot of our genetics, genetic diversity, I'm going to show you a very short, very ghetto video on what is causing a part of the problem that we are seeing. So if we imagine that Africa is the whole box of Smarties, right? It's all the colors. And that's really where human genetic diversity began. So it encompassed all of it. And then about 100,000 years ago, a small portion of those individuals then led to the out of Africa migration, which then subsequently populated the rest of the world. Then essentially the blue Smarties, everyone who went sort of left of the Caucasus Mountains, then developed that European ancestry. And that ancestry is what we predominantly see being used for clinical trials. They're almost cracking up to 500,000 clinical trials, to the best of my knowledge. This I took off the website yesterday. But then when let's look at the proportion of those trials that are taking place representing Africa, and you can see that the number is vastly disproportionate to the population. Not just the genetic diversity, but the population itself. And obviously we know that there's associated with this a disproportionate burden of disease. So what are the consequences of this? If your clinical trials are taking place on a largely very narrow genetic population, what does that mean will happen when those drugs that do work, when they're brought back to a region like sub-Saharan Africa, what are the effects of that? Well, we know that as many as 1 in 12 individuals are admitted to hospital in South Africa, not because of the disease, but because of the drugs that they are taking. And this is being replicated with several studies. And so the issue here is that, and what we're talking about is increased hospital stays. Obviously, it leads to the most severe side effects, which can be death. But it has a huge impact, socioeconomic impact as well on family structure. If you've got people who are sick for much longer than you would expect, not being able to bring home income in an already resource limited situation. So this is what happens as a result of the clinical trial dispersion. So how can we've got this situation where we have the sort of end of the drug pipeline, but we also need to understand what's going on and how we use advanced technologies to understand global health research. Where is the diversity happening there? And of course, we all got very excited 20 plus years ago with the Human Genome Project being sequenced. But I think this graph really says it for me. When we look at genome-wide association studies, GWAS studies, and we look at the individuals in the millions who've been included in cohorts where they've derived scientific information, identified perhaps novel drug targets, you can see that an explosion of these GWAS studies as the technologies become cheaper and cheaper. But again, they're primarily taking place on a very small portion of the population. And for those of you who are looking for the African GWAS studies in purple, if you look very carefully here between the green and the orange, there's a little sliver over there. So certainly for me, genomics research is failing on diversity, particularly as we now understand that these technologies are becoming cheaper. So there's no reason why we shouldn't be able to play catch up in this particular regard. So what does that mean when it comes to gene therapy clinical trials? Well, I just want to bring up two perhaps classical diseases, cystic fibrosis and Duchenne muscular dystrophy. So let's look at the background heterogeneity of cystic fibrosis as a first off, for example. So if we look here at a paper published a few years ago, the authors here did a really beautiful job of looking at the different mutations that existed in different parts of Africa. The mutations that they've listed in red, those of you who are familiar with cystic fibrosis will recognize this mutation here, the delta 508. Now, this particular mutation is the most prevalent in European populations, upwards of 80%. So a lot of clinical gene therapy trials are targeting that particular mutation. But other than South Africa, if you look elsewhere around Africa, you'll notice that this mutation is by no means the most common. It actually often isn't even picked up in countries in Central Africa. And there are lots of other mutations that are coming through. And the difficulty with this is, and we take nothing away from the extraordinary work that CF scientists are doing and moving towards these gene therapies, but there's a difficulty here because those gene therapies are going to be developed and then they're going to apply to a minority of that patient group in Africa. Similarly, if we look at a situation like Duchenne muscular dystrophy, again, you don't need to go into the details of what the mutations are, but you can see in particular, there's a dearth of knowledge in Africa as to what types of mutations are being detected for Duchenne's. And that has implications for the extensive extraordinary amount of work that's being developed towards these two really classical monogenic genetic diseases, where for a lot of the work that's being done, they're really at the forefront of gene therapy trials. So we've got this disconnect where this incredible work is going on elsewhere around the world, but there's a concern that it's going to have limited impact here. And so I just wanted to finish off with one slide where I think this is certainly where my mind is at, where I can see that there are people researchers who are starting to think, what innovative tools can we start using, which can help increase diversity in terms of gene and cell therapy. So first of all, it's really important to note that the FDA has really clocked onto this and increased clinical trial diversity. It's been a particular angle that the US president has been looking at, the Cancer Moonshot goal of making sure that this is being included in clinical trials. So that's very exciting that headspace is starting to clock onto that. But something else that we at my team certainly get really excited about is this, in the sense that where the FDA has just recently approved its first clinical trial using efficacy data that came from cell-based assays. And why is that so important? Why is it important that they've bypassed the animal studies? With increased physiological relevance of the cell-based assays, we can now start enforcing the idea that if we can include cells of diverse origin from African individuals, that these cellular tools can now be used to be incorporated in the early clinical trial, early drug trial development, so that by the time they actually get back to the African population, that acknowledgement of the background genetic diversity has already been taken into account in early genetic trials. So I'm going to leave you with that and hand back over to Dr. Masumi.
Thank you, Janine. Thank you, Jennifer, again. I think to the audience, it's probably clear now that we've been dealing with the molecular part of this equation. So that creates a bridge to go into the public health and the even population genetics part. I'm tempted to start asking the questions with Janine and Jennifer now, but I'm going to hold my tongue. I can see that my friend Judy Coates is egging me to break my own rules, but I'm not. I'm going to invite Dr. Nikane Ndembi to give us the third presentation. And
thanks. Again, thank you for the invitation to the Africa CDC Africa Union to really address this important topic on clinical trials and the diversity. Very important, so that we as an agency of my network, we as an agency, we pay closer attention to diversity, having to deal with 5 Africa Union regions on the continent. Second, having to deal also with different pathogens, and that's a priority to us, including other components. So from the Africa CDC, I'm just going to touch base on how do we build capacity to be able to address some of those really this important topic around clinical trial diversity. I think this is a very important point. If you look in terms of evolution, it's not really a convergent evolution, it's really a divergent evolution over time. If you look at it in terms of ancient history and really gene speciation over time, and also recent history migrations across the globe, I think it's quite important that this topic is being considered. Let me look into just one or two cases, maybe one for HIV. For those who are familiar here, I think we have over 100 strains of HIV. I think usually commonly when people look at HIV, they might assume that it's a single virus. No, it's a huge diversity there across the different strains. And One of those that actually have been really striking, it's some signatures of specific strains that we've seen, or mutations across some of these HIV strains that are actually unique for the entire continent. You can link it probably to HLA typing. You can link it to other components that are actually quite complex to be able to explain those ones. But again, that's a little bit the complexity. We have also observed in the past at least 2 decades that some of particular strains, HIV group O, at some extent HIV group 2, are really confined to some of the regions across the continent, have never been exported at some extent, but we've limited really exportation or spread across the globe. I think those are the questions that we start asking ourselves, what actually the mechanism on being some of these really findings. The second one, I think one of the panelists touched a little bit based on that. We have seen hypersensitivity to some of the drugs like abacavir, which are HLA type and specific, driven by HLA type, and also found primarily among African. I think those are the questions that's very important to look into, as some of the clinical trials at the early inception, didn't include the African population. And those drugs have also been used over, I think, abacavir, and I think because they were discontinued at some point due to the hypersensitivity, they were not actually, trials were not really, the population in Africa were not included in some of these studies to be able to pick them up at the pharmacovigilance of phase 4 trials. So I think, again, if you look at the tremendous work that has been actually achieved through the H3 Africa, which the plan is really to be able to sustain it for at least the next 2 more decades or so, we do have enough funding now to be sure that even within the regions, across the on this continent, there seems to be at least differences in terms of genetics makeup that could actually contribute an impact on the way we perceive some of these key components on genomics. So from the African Union and the Africa CDC, what we've tried to do was to see how we can build capacity across the 5 regions to be able to undertake some of these trials and provide enough information. As you know, some of these trials that are currently being conducted or have been conducted are project-based, and we want to be sure that there's a sustainability over time to be able to generate enough information. So the Africa CDC, we have been able to establish what we call the Pathogen Genomic Institute. For now, the first really impact has been on the genomic surveillance for SARS-CoV-2, and now we're moving towards other pathogens, and including also studies that will actually be able to provide enough information to have a strong database around genomic and genetics across the 5 regions. If you look at this slide, looking at the data that we have so far gathered on the 68 clinical trials, focusing primarily on legacy and expanding diseases, you can see clearly that the Northern Africa will still have a huge gap there. Data or information across diseases are primarily based in the East, West, and Southern Africa. I think Central and Northern Africa, we still have quite enough to do there, and we're putting enough resources to be able to cover those regions as well. I think this is going to form the backbone to be able to generate enough information. I think I'm referring to the NGen sequence, the NGS sequencers that were placed in the 46 of the 55 AU member states. And the plan is really to be able to drive some of these projects, including those that have been, we've referred to the East Africa on this call. So in conclusion here, if you look at this map, what we plan to do is to establish 5 at least reference centers for clinical trials, for R&D, and then also talent development to build that workforce to be able to generate enough information moving forward around the 5 regions. If you look at it, Our target is here in Egypt. I think if you look in the overall clinical trials that we have so far been able to carry out on the continent, 3% of those clinical trials are in Egypt, only 3%, and then 17% of preclinical trials. If you look at Mali, for example, 80% of those clinical trials around across the 4 clinical research centers in Mali, and then 1 in Central Africa, in DRC, and primarily the purpose there is to be able to address some of these outbreak diseases. But again, as I've said, we ride on it to build really a map of the genetic variants that can actually support some of these clinical trial research. And then in East Africa, Kenya and Uganda, obviously in Southern Africa, we're looking at South Africa. I think this is the only way, rather than having some helicopter research here and there, how the continent can respond to allocate resources to sustain genomic studies over time to be able to address some of the public health concerns, as I've said. I think it's unfortunate that some of these trials, they have to be actually carried out in the north and the result back to the south. And this is not what we want to see, because this doesn't help us in terms of vaccine hesitancy, for example. If you look at it for COVID, before I land here, less than 1% of the clinical trials globally were actually conducted on the continent. But all these vaccines that we have from about 10 different manufacturers, none of them actually included the population, the African community, to be sure that they actually take it and lead with it, and then also being able to accept those ones as part of being part of some of these studies. I think this is the way to go: build capacity, generate data, be sure that we attract and build clinical trial sites that can attract researchers to be able to undertake some of these studies across the continent. We cannot continue to blame the rest of the world if we don't build capacity for these 1.3 billion people that we care on this continent. Thank you. Back to you, Dr. Sami.
Thank you so much, Dr. Ndembi, for an insightful presentation. I'm sure our audience has a lot of questions to ask. I do not want to take Professor Mshabela's time, and I'd like to invite him to present the final, to provide the final presentation for today's webinar.
Masumi, greetings everyone. I just want to also start by thanking Amgen for the invitation and Africa Bio. I have no conflicts of interest that I'm aware of. I am Masumi Shabela at the University of KwaZulu-Natal. I'm a professor of public health and Deputy Vice Chancellor for Research and Innovation. I'm not a geneticist. I don't do any genetic studies myself. I don't do any genomic studies, but my job is to create an enabling environment for our scientists who are doing genetic studies and genomic studies to succeed in doing their work. And largely, making sure that they've got the right infrastructure, the resources to do the work. But as a public health scientist, I am very much interested in how this work translates into public health benefit. And I tend to think that when we do this work, we need to start with the end in mind, looking at the public health benefit, working backwards to making sure that we are designing the right intervention, we are testing them the right way through the right processes in order to ensure ultimate uptake. And I'll make a few comments on that. This is going to be my main slide talking point, in the sense that at the moment, in the past year or so, I've been preoccupied with this valley of death. The fact that for a country like South Africa, as Dr. Nike has demonstrated, there's a huge capacity to do laboratory science and laboratory-based research, and in many instances, even do product development. But translating this into something that can then go into full clinical trials and become commercialized is very difficult, because we have this huge gap where when it comes to preclinical studies and pilot testing, optimizing the products and optimizing active pharmaceutical ingredients and so forth, we are not able to do this, largely because we don't have the right infrastructure. We don't have the relevant small scale GMP laboratory facilities to do work. Now, why am I preoccupied with this? I think that lessons that we've learned from COVID and from many of the clinical trials we're going to have to do going forward is that we need to ensure that we're building a complete value chain. Otherwise, this problem that has been raised, that products will be developed elsewhere, sometimes tested here and sometimes not even tested here, but because of their success elsewhere, they will have to be adopted and be used as the only options available, cannot be overcome without building a full value chain. And for people like myself who are comfortable in being generalist, we do want to work with people from different disciplines to connect all the different dots. We want to work with government to make sure that they support the necessary building of the infrastructure, they inject enough investments into the infrastructure. Not only that, but also the development of the talent that is necessary. It's important that we work with the different research institutions, councils, to make sure that we build this value chain and the full value chain. It's also important that we work with regulatory bodies, as Jen has also mentioned, the importance of regulatory bodies in ensuring that the work that is done here is done in a safe manner. And safety is very important, because we have seen that if we don't ensure safety, a lot of misinformation can emerge in terms of the integrity of the products that we develop, even if it's gene therapy. And if people are going to, for example, provide evidence of errors that happen in this phase, it can contribute a lot to mistrust that we already have at very high levels in our society currently. Therefore, a degree of transparency around exactly how we are going about testing these therapies is going to be really important. And I think that as we do that, we also have to do it in a way that, as already mentioned by Jen, that engages with society and communities early on, not necessarily after the fact, not only after the fact, because if we do it after the fact, then we are not necessarily going to take people along with us. So this, for me, is really important, having learned from COVID that we need to build diversity in clinical trials. We need to build diversity in terms of biobanks and African genomes and genetic studies. But we have to do this cognizant of the full value chain from the beginning. One of the successes we have seen in South Africa, and really much of Africa, including what we've seen in Botswana elsewhere, is the success of the sequencing work that has been done around COVID-19 variants. And really the success of it came from different institutions coming together and the partnerships. And I'm really interested in us thinking around genomics diversity and genetic diversity and the diversity of clinical trials, in what sort of partnerships are going to yield success for this work. Because if we are just going to look at it as something that has to only be done within the discipline, it's going to be very difficult to reach the kind of level of partnership that will have leverage to ensure impact. We know from the lessons that we have on the African context that it is when different partners come together that we see a lot of success, and that kind of success can have a global impact as well, and can produce world-leading science, as we have seen already. And that's what we have to aim for. we do need to work with partners overseas and abroad. This is very important. But those partnerships also have to be governed by key principles of co-production, just because we do know already that, as we have already seen, that some of the countries in the north are already leading when it comes to gene editing and gene therapy. And this will contribute a lot to hierarchies and unequal power dynamics that we're going to have to manage. And for some of us, in my role, I am concerned about these kind of issues. When we are going to do capacity building and strengthening, how do we do it in ways that are mutually beneficial? When we are going to set up infrastructure, produce data, produce sequences, produce samples, how are we going to do this in such a way that it will actually respect the rules of engagement between northern and southern partners? As we have said, we have learned these lessons in global health, that these dynamics can contribute to a lot of inequity. And of course, we do want to make sure that we build capacity to also analyze data on the African continent, and we don't always have samples taken elsewhere, including having to build capacity to store those samples here. And because of the fact that a lot of this work is informed a lot by training people in STEM and building the strong foundation in STEM, we have to do that work when it comes to us as universities. But most importantly, we have to make sure that the education will start to very quickly include social science and humanities aspect. Just because, as we have seen, you can develop a product that people are not going to take, and a lot of engagement with communities really require participatory methods that our laboratory scientists may not have experience in doing. But if they partner successfully with people who work in the social sciences and humanities, we may succeed in making sure that we build seamless connections between what happens in technology development and innovation in the laboratory and in the development of those products in relation to what happens in community and society, including when we have to engage with governments and industry. I want to finish off by saying this, that for me, I want to take all of the work that we will be doing here in building capacity for biobanks for African genomes and infrastructure for genomic studies, and increasing our own capacity to do clinical trials, that we have to really see it in the context of our society, not only from a genetic diversity point of view, but really also from a societal impact. As colleagues have shown, that there are certain diseases that if we demonstrate early on that we can show impact on them and public health benefit for them, that public health benefit will provide an incentive for buy-in from communities, buy-in from governments, buy-in from industry, but also buy-in from the gifted and talented young scientists who we want to recruit in terms of training and development along these value chains that we want to build. And also just making sure that we can get support to build the necessary infrastructure. At the end of the day, the strongest argument from my point of view will come from the public health benefit perspective. Let me stop there, Dr. Masumi, and thank you for affording me the opportunity, and the listeners as well. Thank you.
Thank you, Professor Mashabela. I think that was a very good way to tie everything together before we move to the questions. So there are two types of questions. There are those that have been typed onto the chat link, and there are others that I will use my prerogative as a facilitator to embarrass the panelists. Luckily, I've already forewarned them to say that brace yourself for my famous questions. And I think in the tradition of the fact that Jennifer, again, happy Thanksgiving, Jennifer. I know that you're in the US and it's Thanksgiving today. Happy Thanksgiving. So the first question is directed at you, particularly because you spoke about gene therapies. And one of the, as I said, as I've always mentioned, one of the fundamental or founding tenets of BioAfrica is the realization that biotechnology is just over a century old and it's experienced a number of patent cliffs. What it means that the space has been democratized and that intellectual capital has now risen to a point where continents like ours, if we do the correct thing and we have the correct strategy, can access these therapies. And the biggest gripe that we have are issues around, for example, the fact that almost 41 years after HIV was first characterized, we still don't have a vaccine. But 3 years after COVID came about, we had a vaccine. And that was largely because of the distribution of resources, north-south. And I'm just wanting to provocatively ask you whether you think gene therapy has a role to play in low and middle income countries, and how would that work? Just your thoughts to address some of our intractable challenges, health challenges, from a product development perspective.
Yeah, so I will say, I think the short answer is yes. I think gene therapy could have an incredible impact in all countries. I think it's been about 30 plus years in the making, and the approvals that I talked about are the proof of concept that there is a lot of therapeutic potential in these approaches. Maybe not for every single disease, but certainly for a large number of diseases. For example, there's a lot of the approvals are in the space of cancer treatment, as well as in the treatment of inherited or genetic-based diseases. The infectious disease space is just starting to see some fruits, but it's taken longer to get there. So likely for sickle cell disease, which is an inherited disease, there are gene therapies that could be useful now. As I mentioned, one of the approved gene therapies in the US has applications in that disease. For HIV, we're still working, right? You saw there were 3 times the number of clinical trials. It's been 2 more decades, and we still don't have an approved product there, so it's taking us a little bit longer to learn. That said, there are really important steps that we have to take. I think Dr. Moshibella said this so well. I cannot stress enough to how important it is to talk to the people who will eventually be participants, and to work with social scientists and socio-behavioral scientists. I give this example all the time. It's a famous example in the United States of a muscular disorder similar to the DMD that Janine described, where researchers spent almost 15 years developing a genetic therapy that could be given to the heart and to the diaphragm, because most of these patients die of heart failure or the inability to actually control their breath. And they brought this to patients, and they couldn't get anyone to enroll in the trial. And when they asked patients, why don't you want this gene therapy? Patients said, I don't want to live another 10 years with a colostomy bag. Can you please make a gene therapy that gives me the quality of life of being able to control when I go to the bathroom? And it was a huge moment where the realization between what we thought people needed and what people actually wanted was very different. And so this is just one reason why it's so important. All of gene therapy is still in its infancy, and there are risks that we don't yet know because we haven't treated a large number of diseases, and we have to make sure that people are on board to understand that. So I will stop there and just say, yes, I think gene therapy definitely has a place. I am a firm believer that it should be developed by the people where people are going to be treated, not by people like me and then delivered to someplace else.
Thank you, Jan. And I think that opens the space for Janine to be put on the spotlight. You had a last slide which spoke about cell-based assays. And I just wanted to just tease some of your thinking around that, how it is potentially in a groundbreaking intervention or technology to push the envelope and to push the boundaries of doing clinical trials in this continent, particularly thinking about what Professor Mashabela showed as a value of death, preclinical and all of those things. I just want to, and unfortunately, you only gave us a snippet, silly you. If you could just maybe just take us through that and to try and maybe answer some of the concerns that were raised by Professor Mashabela.
Yeah, sure. Thank you. I was trying to keep to time. Look, there are multiple reasons why the cell-based assays are increasingly becoming valuable. Compared to preclinical animal models, they are a lot more inexpensive, a lot less expensive, essentially. So that's number one. So you can do a lot more on a very small scale and learn a great deal more. Obviously, they don't have whole physiological relevance, right? You don't have the in vivo response of what's going on. But what you do finally have is the opportunity to understand the response in a human genetic background that's not just of one person, which is what a lot of the pharmaceutical companies have been using, one or two cell lines that they've been testing drugs in. It's now becoming increasingly obvious that those pharmaceutical companies and others, researchers around the world, are not confident about their data because they've only been using a very narrow one or two lines. But if you increase the diversity of the individuals from which the cells come, you just as a scientist, you have more trust in the assay that you're working with, because it's almost like you're doing little mini clinical trials on large numbers of individuals, for one. The other thing that's really important about these cellular assays is that, and I didn't have time to talk about them, but they are very malleable. Depending on which kind of stem cell work you do, you could turn them into liver cells, you can turn them into cardiac cells. So they're really, really adaptable in terms of an innovative tool for preclinical development. So there's that. But I think one of the things, an example, and again, Jennifer did this really well with one particular example, and this really blows my mind. So I wish I'd had time to talk about Hans Cleaver's mini guts, for example, for cystic fibrosis. And essentially what they were able to do there was take, it's not sort of the top of the list stem cell, it's sort of halfway down, but you have little stem cells in your gut. And they were able to take tiny gut biopsies from individuals with cystic fibrosis, and then sort of digest them up and then grow little mini gut organoids in a dish. These are so small, you can barely see them with the naked eye, but you can make thousands of them. And what you can do with that is you can turn it into an assay that you can screen drugs on that and say, does this drug make this, improve this calcium channel, for example? And you can actually see it. They're enlarged like this. It's the sweetest little thing. So it's an amazing experimental tool that can be done on a person-to-person basis. But let me round off to my point. One of the things I spoke about is that for a lot of these gene therapies, it's often targeted to one particular mutation. Now, with an example like this, what you could do is you could actually whittle down patient groups into what they're calling serotypes. So you could find one drug that might suit a whole lot of mutations in sort of bucket loads. And that obviously is going to massively reduce the cost. And then the valley of death of trying to get something that's perhaps targeting one particular mutation through the myriad sort of hoops that it has to get through to be approved at the end is difficult and it's expensive. So if you can create little shortcuts earlier on, that's where I think it's going to be making the biggest difference. That's where the cell-based assays come in.
Thank you. And I think I'd like to invite Dr. Ndembi maybe to come in here, and I would ask him a question once he's made a comment in terms of what Janine and Jennifer have said. If you're available.
Yeah, Dr. Somi, good discussion. But for the layman person, probably on this call, I think the question is, do we have policy and resources on the continent to move to that pace now? That's the biggest question you want to address from the public health perspective. Me sitting at the African Union and having to see requests from member states, 55 member states, do we think that is the way to go right now with precision medicines, where we don't have vaccines just for malaria was a few months ago. Dr. Somi, you mentioned HIV, we still have to deal with it. Tuberculosis, we have thousands of outbreaks across the continent. My question will be, do we go for inclusivity to be sure that we advocate for more clinical trials where we have data from this continent to support that this will work or not for a particular drug designed somewhere and a generic now for the continent? I think that's a big question that I have for my colleagues, panelists, to be just pragmatic. So that's my question. And I know, probably correct me there, that South Africa is probably the only African country with policy on precision medicines. But other countries on the continent, I think we don't have enough data. Maybe that's my initial comments there. Without being, I'm from the genomics background, I can tell you. So I like fancy stuff. But with my experience with the African Union and public health, is that how do we bring that to the community? That's the biggest question. Over to you, Dr. Somi. Somi?
Thank you. You're not going to get off so easily. You presented to us the immense work that Africa CDC is doing in building capacity across the continent. And I guess one of the questions that one wants to know is whether the data that you're generating is going to be available for the researchers in the continent and globally, number one. But number two, whether the strategy or the program that you are currently pursuing around building capacity is forward-looking. It's not being done on the basis of what essentially is. And that's why I mentioned the issue of patent cliffs, that if we are going to catch up as a continent, we best embrace the technologies of the future. And that when you build a capacity, you're doing it with that in the mind, instead of doing the same old, same old. Just a provocation from me to say, what is your sense in terms of doing that? And I think Professor Michabela, don't relax, it's coming to you in a second. But DK, maybe if you could just answer us in terms of the integration of these advanced technologies, gene therapy, it seems to me cell-based assays, particularly if you consider that a lot of our challenges are around epidemics that are localized, and that where there is no incentive in the global north to invest in developing solutions to those. Back to you, DK.
No, absolutely. Thanks, Dr. Somi. I'm sure also Mashabela will come here as well. But the bottom line here, Dr. Somi, as we speak, I think to be quite really put it in the picture, we have over 15 outbreaks currently on this continent, 15. And we have to deal now with COVID, we have to deal with Ebola in Uganda, and other outbreaks, measles, and all those that you know, 15 outbreaks, and undergoing. Some are really we don't really blow the trumpet around those ones because they're being contained. And some small as cholera, for example, that has been eradicated years and years in other countries in the West, we still have to deal with them. Over 20 countries are experiencing those outbreaks. But the bottom line is that we need to invest into genomics. It's clear, you know your pathogens, you can deal with the disease. So now the point here, that's where we started. Where do we start from? I think clinical trials is the tail end of it, where we cannot even diagnose. We have seen what happened during COVID. People were carrying rapid diagnostic tests and PCR tests in suitcases. We cannot do that. We can't continue to do this. So if we build that capacity, is to be able to have a larger database anchored and actually we can share across scientists across the continent and beyond the continent to be able to just design rapid diagnostic tests or primers for a PCR. We've seen what's happened. Only one country another South Africa and probably Senegal at some extent were able to diagnose COVID when it started 2 years ago. Just basic. So I think it is important that we start investing. I think that's where that workforce development from Professor Mashabisha, I love it because we need to build capacity to be able to do that. The straightforward answer, Dr. Somi, yes, data are going to be made available, the same for H2 Africa. They might take some time to ensure that local scientists, they customize it, they can be actually do some little bit of translational research there, design target for rapid diagnostic, for therapeutics and vaccines, and then we open up for the rest of the world. I think that's why the data is primarily being built for. I think, again, that comes in an environment where data sharing is now being discussed extensively. WHO released the guidelines, African Union released as well. How do we now be sure that people consent, at least that we share those information at some stage? I think that's the ultimate goal for a public health agency, Dr. Somi. Yes, is to share and then be sure that we move science and being self-reliant in the near future. Thank you. Back to you.
Thank you, Dr. Dembi. Now the spotlight is on Professor Mushabela. And I know it's payday tomorrow, so I'm going to try and be gentle. You sign my check. But the question that I want to ask you is that it's all well and good to talk about the value chain and the value of debt. It seems, at least to me, the uninformed me, that one big part of the problem is the role of public health. You're a professor of public health. The role of public health in shaping the agenda. Now that we have all these technologies, all these exciting innovations and developments, what is the role of public health in ensuring that the research or the product innovation that is being done is directed towards the real problems, as opposed to where the markets are, the lucrative, highly profitable products are? And how could this be bridged?
Yeah, no, thank you for that, Dr. Msomi. I think that what I think it's really interesting is that the contribution of public health as a field is not necessarily only going to come from the public health scientists. Actually, it's going to come from a combination of people that come from different disciplines who are interested in the overall public health benefit. And let me give you an example with the fact that, as you were saying, with HIV and TB, actually, we found that there are people who are dedicated to doing that work, doing the basic sciences, doing the clinical research and so forth. But they are often a sort of community that's working on their own. And they can work every day and nobody really sees what they're doing. Government was not paying a lot of attention. Industry was not paying a lot of attention. But when COVID came, because there was such a huge public health benefit that people stood to gain, we saw a lot of people, even in other fields, pivot to actually direct efforts towards COVID-19. One of the things in South Africa that was really interesting was the involvement of the interface between public health sector and private sector. For me, that was really interesting, where we saw a lot of innovation, a lot of expertise in the private sector coming out to actually support the effort. And in fact, some of the work that we did on sequencing, where we picked up the S gene drop that we saw with the variants, came from the private sector, and they notified the public sector. And for me, that's the thing. Like, I'm very keen to see us get to a point where we can agree that a condition is a public health priority, and it's a priority for everyone, not just for public health scientists. And when it becomes a priority, then we are able to draw on a range of our expertise to actually drive a lot of effort in finding solutions. I would really like to be able to say to, well, actually, 2 days ago, I was speaking to our HIV pathogenesis group. They've done a lot of work over a period of 20 years. They've done amazing work. A lot of the HIV research that has come out of KwaZulu-Natal is incredible. But I was saying to them that, look, for me, I would like to point out the problems that we have, set priorities with you, and you go into the lab and you come up with solutions. And when you've got candidates, products, and potential solutions, I will work with you to make sure that those products are tested. I will work with government. I will bring in industry to try and find ways to actually get those products optimized and moved in, tested, and eventually moved into the market, onto the shelf. The problem is currently people work in silos, especially in South Africa and in Africa. And the second problem that I see is that when people are interested in pushing their boundaries, they leave the continent and they go and work in other northern countries. And that talent is lost to Africa as well. And this thing of brain drain is really hampering the progress that we could see on the continent in terms of making sure that we see the public health benefit. But I do think that there is benefit to brain circulation, where people do get exposed to different technologies and techniques and methods elsewhere in the world, and they are able to come back on the continent and help kind of develop products and advance research. So there is benefit in terms of brain circulation and also in terms of mobility, but partnerships as well. It's just that we need more of our African scientists to take the lead in setting the agenda that aligns with the priorities that we have, so that we can develop the products that are relevant to our priorities and develop them in a way that works best for the communities that we serve locally. And if we really do proper community engagement in that process, we can also get buy-in early on. And it would be really good if we can get this sort of lead scientists from Africa to get other partners from the global north to prioritize the problems that we have, and for them to contribute their expertise, and for some of them their resources to the problems that we have. We shouldn't be struggling with 15 outbreaks right now, when we already have demonstrated that there is potential to be able to effectively intervene if we intervene collectively. But that's the sort of thing that I think we need to move towards. How we do it when there is so much competition across the world and so much inequity, that will be a challenge that we will continue to face. But we have learned lessons in the past couple of years that can really help bring that whole value chain to work, as long as we focus on driving a common agenda around public health benefits. Thank you.
Thank you, Professor Mashabela. I'm told by the production team that we've only got 10 minutes. So I'm going to request each one of our panelists just to give 2 minutes final, but I will ask the question. And I'm going to start with my famous provocative and controversial question. I'm getting some looks from my friends at Amgen asking me, when is the controversial question going to come? I'm glad that it doesn't come from me this time around. It comes from Yusuf, one of our eminent panelists from previous webinars. And the way I frame it is there's this Netflix documentary that talks about the inherent bias in artificial intelligence in terms of facial recognition, and how people end up being mistaken because when they do the coding, it does not include non-Caucasian features. For example, I guess the same question that's been posted here is that the chips that we talk about in cell-based assays are not developed by people in the continent and are predominantly focused on white Caucasians. And how do we build capacity on the continent to have African scientists and engineers and leaders in this space to make these cell-based assays more context and more effective for what we're trying to achieve. And I think I'll ask Jennifer and Jenan to answer that question. We have two more coming from Nikkei, and I promise to stick to my famous controversial nature.
Jennifer, do you want to go first? Do you want?
Sure. So I would say, in terms of the core question, which is like, how do we increase, access on the African continent? I don't think there's a one-trick pony answer here. I think you need a lot of different ideas and a lot of different attempts and different strategies, and then we need to look at what is working best. So I will tell you some of the things that we're trying. Again, coming from a place where I firmly believe gene therapy to treat Africans should be developed by Africans. How do we encourage that development? So step one would be transfer knowledge and technologies. And the way we can do that is by having short-term training where we have visiting scientists come to the US. We've done this with nurses and doctors who are on the clinical side of the gene therapy treatments, and we've just done it with scientists. A little plug, we have our first gene therapist who just got 2 years of funding from the American Society of Transplantation and Cellular Therapy for starting a gene therapy group in Kampala, Uganda at the Joint Clinical Research Center. She actually just received that award this week, which is fantastic. And so, she spent 2 years in my lab learning how we do gene therapy for sickle cell and HIV, and then she's taking that back and she'll have another 2 years to get that group started there. And I'm sure we're going to need more runway than that. So we'll be looking at lots of creative strategies to encourage funding for new institutions, naive institutions across lots of different countries in the future. And so, one of the things we have right now is you talked about siloing in Africa. This is the same is true. Like the majority of the research funding in most countries is restricted to people in those countries and does not allow for a lot of cross-continental collaborations. And so we definitely need to think about how to change the funding structures to allow partnerships that allow us to share that information. And I think really the way that we can inspire people to have lots more ideas than just those is to, remind them that if we can all do this together, the more brains in the room, the more likely you are to have a sustainable and accessible solution. And I often say, the strategies that individuals in low and middle income countries have had to employ to solve problems of distribution are things that will actually benefit the entire world. I talked about the US being the majority developer, but we're not getting it right here. If you look at our national distribution of patients with HIV, the gene therapy trials that are being conducted for HIV are not happening in those geographic regions either. It's mostly in the southeastern United States, and the majority of the work is happening on the West Coast. So we're not even able to distribute within our own country very well. And so we also need the solutions and more brains helping us solve that problem.
Thank you. Janine?
Yeah, so Jennifer said a lot of what I completely agree with. That training, I was lucky enough to have a postdoc that sent me overseas with a specific mandate to go and learn and come back, and that's where I picked up stem cell technology. It was verbatim what they required. But the thing is, and this is, this question about, for example, the microfluidic devices, they're not that complicated, right? They're a little bit, but they're not that complicated. But everything costs more and takes longer to get here. So it's not that it's, if you had equal amount of money, it still makes it difficult because everything takes so long to get here. Primers cost 10 times the amount, and those are basic things that we need. So I'm actually going to turn that question on its head. And one of the things I've noticed with my team in particular, because we can't run with the same expert equipment that some people do in Europe and the States. And I watch my team do it, and they MacGyver a lot. So they, you know, there's duct tape and there's drilling holes in shot bottles, and they completely turn a decommissioned cardiac bypass machine into something that can cannulate and digest specific cell types and organs, tissues. They do amazing work. And I guarantee you right now that if someone gave my team the same amount of money as a team, and Jennifer, I take nothing away from your extraordinary researchers, but the same amount of money, I guarantee you they do double the amounts, because they have to think so carefully about what they do. So I know it's frustrating, I hear what you're saying, but there are advantages to doing it. And I think often we think really hard about our research, and we can get a lot more done. So turning it the other way around.
Thank you. I'm hoping to invite you to a future one. Don't congratulate yourself. You have not gotten away scot-free. There'll be a second chance. I think the last question, again, I'm going to be taking it from the audience. It's directed at Dr. Ndebi and Professor Mashabela. And it's a question, I'm turning it around. It was originally posed to Jan, and was around collaboration with African institutions. And the question I want to ask is, we talk about collaboration, and Professor Mashabela was quite emphatic in talking about brain circulation. What I want to know from Dr. Nike Ndembi and Professor Mashabela is that Africa is not a homogeneous place. It is not the same in terms of development. In the work that's being done, how much of the intra-African collaboration is happening in between the places where there's advanced genomics? There is a potential for, Jan spoke about work being done in gene therapy in Uganda. I'm just trying to understand what are the linkages between different African countries. And because Dr. Ndembi spoke about regions from an Africa CDC perspective, and and Prof. Mashabela, you work at a university, so your primary responsibility is to produce these skills. What is the level of collaboration, particularly to exploit the opportunities that come from collaboration? Over to you, Dr. Ndembi, and then Prof. Mashabela can close off for us.
No, I think, thanks, Dr. Somi. Yeah, I think there's now, I think that's probably now trends across the continent that scientists are really engaging in grant writings across the different regions. When we started the DART clinical trials, one of the largest clinical trials around antiretroviral therapy in Africa, I think we started by engaging the different regions, for example, getting Zimbabwe, getting Kenya, Uganda, South Africa. That's the trend now. Well, because we clearly understood that you cannot do that in a single region and generalize it for the rest of the continent. Very important. And what I've said on the PGI, for example, and which is the role of Africa CDC, coordination, really, that's stewardship to ensure that we have the whole of Africa approach. For example, for Pathogen Genomic Institute, genomics platform, we're having next week in Addis. I think basically all the institutions working on genetics across the continent are going to meet at the African Union. And some of this conversation that we're having today going to be tabled again. How do we engage collaboratively? Leverage on the infrastructure, the expertise, the workforce, localization, to be sure that we drive these genomic studies across the country. I think that's the way to go. We just have to provide some catalytic funds for some of the institutions to be able to really get engaged and work collaboratively on this, Dr. Somi. I think that's from the African Union perspective. Even as individuals, as PIs, we do the same. the NIH grant, some of the R1, we engage other countries, Uganda, Kenya, part of the one in South Africa. Again, with the structure in our mind that even just the strengths that are circulating in East Africa are different to what you see in West Africa. So we really want to be sure that that inclusivity and also that diversity in our study population is being reflected in the way we present some of our findings from the continent. I think that's what I can say. And my point has always been to really to be able to encourage that dynamics across the different regions, so as to ensure that we can start driving some of the conversation based on our data that have been generated, and see how they are different across the different regions as well. So I think that's my final take there. But again, before since I still have really the mic here, Dr. Somi, I think it's very clear. Malaria, just malaria, you're going to be shocked that we cannot accurately diagnose some of the strain, very basic, the species. And I'm talking hepatitis C, that's the same thing. Even the tests that are being used, some cannot because of the diversity. So I'm just giving, I can go on and on to say there's a lot to be done at really the downstream level so that we can start really understand our own pandemic and all diseases that are actually circulating across the continent. Back to you, Dr. Son.
Thank you. Thank you, Nikai. I think the last question, and I'd like to request Prof. Mshabela just to give us a 2-minute wrap. And I'm going to throw it back at you because you spoke about brain recirculation. So I hope you had thought hard before you used such a big term, brain recirculation. And I'm wondering whether there is an opportunity for this brain recirculation within the continent itself, so that we build these capabilities in the continent through collaborations that are productive and strategic.
Yeah, so let me speak about 2 things, Dr. Msomi. I think the first one is that, well, I mean, I'm fortunate in my job to be exposed to different groups and different sort of diverse networks. And one of the ones that is happening in Africa is called ARUA, the African Research Intensive Universities Alliance. So all the 16 top research intensive universities have collaborated across disciplines, and they run centers of excellence. And one of them is dedicated to vaccine development, and it's got nodes in South, East, and West. And it's the kind of place where we've been negotiating with the partners in the global North. And recently there was a meeting of the European Guild universities. 18 vice chancellors met with vice chancellors in South Africa to negotiate with them around issues of partnerships, issues of brain drain, and mobility, and how to make sure that we don't lose talent on the African continent. And one thing that we have put on the table, for example, it's the need to develop centers of excellence around instrumentation. And it's just about making sure that there is equipment and infrastructure in different parts of Africa that people from different African countries can have access to. If you want a special equipment that you don't have in your country that you want to use for your experiments, you know, through this network, you should be able to go to that university and access that equipment. And that is something that we are pushing for. And hopefully it will not just be a center that is physically located in one place, but it will be a virtual center where instruments are made available across the continent. And so that is something that I think it's potentially exciting. One of the things that we found when we were doing benchmarking studies and looking at issues of brain drain was that people leave also because they are not just looking for expert mentors, but they're looking for advanced facilities overseas. The second thing I want to mention is the data science. You know, African genomes and genomic studies, we have huge data sets that are going to come, and we need powerful computers that are going to analyze this and powerful techniques to do this. And we are lagging behind when it comes to data science in Africa. And we've been collaborating and bringing together groups from statistics and mathematics and computer science with people from engineering and people from physics. And one of the things they raised is the issue of data security, that we are going to lag behind when it comes to data security. And if we are not going to assure people of data security when we collect their samples and their data, very sensitive data is going to be very difficult. So I just want to say that, you know, quantum physics has already made to us that if data is not quantum secure, it's not going to be secure enough. So we're very excited about those possibilities. And I will just leave it there, Dr. Masumi, and say that there's definitely a lot of conversations, and I can hope that we can place African Genome's agenda on the table in those kind of networks. Thanks.
I think with that, we've come to the end of our seminar. These always feel short, and that is why we are looking forward to the 2023 series. What we would like to do is to invite everybody to provide suggestions as to the issues that can be discussed in 2023. We have these seminars generally every 2 months. And I'd like to thank the panelists. This was a showstopper to close all showstoppers. And I'm so glad that I was part of this facilitation. And I have some personal admission to make around gene therapy. When I started doing biotechnology or genetic engineering in the 80s, gene therapy was a pipe dream. And today I met a real gene therapist. And Janine, Nikkei, and Professor Mshavela, thank you so much for making this today's engagement such a worthwhile one and something that we could look forward to the next year. And obviously, these are not just for information. These are opportunities for us to meet each other and to begin collaborating in earnest and to look forward to future collaboration and expose some of our young scientists and rising stars to this type of and this quality of thinking and engagement. Thanks, and I'm glad that we're only 3 minutes late. We were meant to finish at 18:35. So I think to Amgen, we thank you for the support, and we hope that it's going to happen next year. And I think we're not going to be penalized for the extra 3 minutes. Pina and Judy, I do hope that you know where to place the blame, not me. Definitely not Africa Bio or Bio Africa. Thank you so much, everybody. And thanks to the audience for such engaging questions. And as I said, we will be sending the recording of today's session to the audience and to different people together with the previous webinars. And we would like to invite you guys to co-create the future agenda for these webinars. Thank you. Thanks, everyone.